Welcome, and today we are going to be talking about how cells harvest chemical energy. So now that we know a little bit about cell organelles and a little bit about chemical energy, we can put it all together and see how cells break down glucose or sugar and get energy from it. And that is all called glucose metabolism or cellular respiration. So first we're going to talk about what cellular respiration is and compare that to photosynthesis. So what do cells do? Plant cells can do both cellular respiration and photosynthesis, and animal cells can only do cellular respiration. So we're going to talk about that and how that compares to breathing respiration. Why is that different? And then we're going to individually go through the stages of cellular respiration. So how do we actually break down that sugar or that glucose and turn it into energy? And then lastly, we'll talk a little bit about fermentation. And the thing about fermentation is, and how it's different from cellular respiration, is our cells use oxygen in cellular respiration to break down that glucose, but fermentation doesn't require oxygen. So it's how a lot of people are familiar with fermentation, with how to make beer, right? Beer and wine undergoes fermentation. So how does our body actually utilize fermentation as well? So photosynthesis and cellular respiration are both how cells provide energy for life. Okay, so what is the difference between them? So in photosynthesis, they're actually capturing sun energy and turning it into food. And the only things that can do that are plants. So we cannot undergo photosynthesis. I wish we could just use the sun energy to make food, but we can't. We have to utilize energy and eat food to make energy. Okay. So in cellular respiration, that energy that we're actually getting from our food, we turn into ATP. So ATP is our energy that we get from the food, and we can then utilize it in other chemical pathways in our body. So these two reactions have opposite reactants and products. So they're almost like equal and opposite reactions. So let's take a look at that in our picture. So first we'll kind of start with photosynthesis on the top. So here's our chloroplast up there, and the chloroplast is what is actually undergoing photosynthesis, okay? So that guy is going to actually capture some sunlight energy, and we'll talk about that in our photosynthesis lecture. But then it's going to use water and CO2 from the environment and that energy, and it's going to create sugar. So this organic molecule here is food, essentially, for the plant. And another product is oxygen. Okay? So plant cells are able to turn water and carbon dioxide into sugar and oxygen. Now conversely, us, animal cells, but also plants can do this too. So plants are cool because they can do both. But animal cells only can do this. And this happens in the mitochondria. So the mitochondria in the cells are going to take that sugar, that glucose, organic molecule, and oxygen and create energy, create ATP. And that ATP is going to then be used in other chemical processes in the cell. But then the product, the waste products from turning that food into energy, is carbon dioxide and water. Okay? So for the different processes, our products and our reactants are reversed. Okay? So they're great processes. One is producing food and one is producing energy from food. Okay? So we'll talk more about photosynthesis in the next lecture, but what we're going to talk about today is this process of turning sugar into energy. So now you might say, well, respiration is what we do when we breathe, right? So organisms that undergo respiration is breathing, and that's the exchange of gas molecules in an organism. So we're going to breathe in oxygen and we're going to breathe out CO2. Now, why we use the word respiration when we're talking about this process in cells is that's kind of what the cells are doing. They're bringing in oxygen and expiring or having a waste product of carbon dioxide. So that's why we use the word respiration, but it's not the same thing as breathing respiration. So that's happening in our lungs. But what's happening in our cells is we're breaking down food to make energy. So cellular respiration is what is happening in the cells, whereas breathing respiration is what is happening in the lungs. Okay? So a little bit different, but kind of similar products and reactants. So now if we go back to our lecture about energy, is cellular respiration an endergonic or an exergonic reaction? So are we creating energy or are we requiring energy? So go ahead and pause the video and think about it for a minute. So here is a little picture to help explain that, but it is an exergonic reaction, okay? So exergonic cellular respiration. So we're taking food and we're creating energy out of it, okay? So then we have fuel for other processes in the body, other endergonic processes that actually require that ATP to drive that reaction, okay? So cellular respiration is an exergonic reaction because we're taking fuel, we're taking food, and we're creating energy out of it, okay? So we're releasing energy. So I have a wonderful video on cellular respiration, and it helps to explain a little bit about what's going on. And so because I can't link it in the PowerPoint for the video, I have it linked in Canvas so you can actually watch the video there. So go ahead and watch that either now or after the lecture, but I really think it helps to explain the process a little bit as well. So here is an image that is showing you the steps of cellular respiration in a very simplified format. So essentially there's going to be 4 steps to cellular respiration, and we're going to go through all the individual steps. So we have glycolysis, which is essentially the breakdown of glucose. Then we're going to use that breakdown product in pyruvate oxidation, which will then give us a product that can go through the citric acid cycle. Okay, so that's step 1, 2, and 3. And then we're going to undergo what's called oxidative phosphorylation. And that's really where the powerhouse is to get a lot of energy out of it. Okay, so we're going to go through these different steps, but if you notice down here, a lot of these steps are going to be providing energy as we go. But this big oxidative phosphorylation last step is where we're going to get our real big bang for our buck for that ATP. So this is our general reaction for cellular respiration. So we're taking that same reaction that we talked about earlier and just breaking it down a little bit. So here we have the chemical structure for glucose, which is C6H12O6. Okay, so carbon, hydrogen, and oxygen. Then we're going to take oxygen, right, O2. We're going to mix these two, and we're going to get essentially carbon dioxide, water, and energy. Okay, so what's actually happening? This is a redox reaction, And I know maybe some of you aren't familiar with redox reactions because it's more of a chemistry term, but essentially it's what's happening with the electrons of these molecules. So the cells are actually going to capture the energy when the electrons, quote unquote, kind of fall off of the organic compound such as glucose, and then we're going to give it to oxygen. Okay, so that's what's happening. You're kind of transferring energy, because remember, we can't create energy. First law of thermodynamics, energy is neither created nor destroyed. And when you transfer that energy, you're going to lose a little bit of energy along the way. And guess what we lose that energy as? Well, that's heat. So we are thermoregulating by all these metabolic processes in our body. We're giving off heat, right? We're like little powerhouses. So when that glucose is going to be turned into carbon dioxide, we're losing hydrogen atoms, okay? Which in turn means you're losing electrons. So you're becoming oxidized, okay? Leo says grr, remember that, okay? And we'll talk a lot more about that, but losing electrons means you're being oxidized. Now the oxygen, on the other hand, is gaining hydrogen atoms, meaning it's gaining electrons. So if you're gaining electrons, you're being reduced. Okay, so it gets turned into water. And by moving all these electrons around, we can get some energy out of it. Okay. So now we can actually produce up to 32 ATP molecules per glucose inside the mitochondria. So again, we'll talk about how we get all those ATP molecules, but that's quite a bit of energy per glucose molecule. And if you think about, starch and cellulose and all those polysaccharides that are made up of all those glucose molecules, that's what we're breaking down, all those polysaccharides into glucose so that we can use that one glucose molecule to produce energy. So we can actually capture about 34% of the energy in glucose. So again, when you transfer energy, you're losing some energy. So we're not gaining all the energy possible out of glucose. We're just taking a fraction of it, and the rest is lost as heat. So here are our stages of cellular respiration. So we're going to go through these individually. But the first step is glycolysis. And the big thing to note about glycolysis is that it's happening outside of the mitochondria. So it's happening in the cytosol, okay, of the cell. So it's happening outside the cell, outside the mitochondria in the cytosol of the cell. Now, bacteria undergo this as well, but it happens just kind of right outside the bacteria, okay? The second step is pyruvate oxidation. So that's the first step that's actually happening inside the mitochondria. And then the citric acid cycle is also happening. The rest are really happening in the mitochondria. And glycolysis starts the breakdown of sugar, but the citric acid cycle completes the breakdown of sugar. Okay, so those first three steps are really all about breaking down glucose. And then oxidative phosphorylation is using those electrons that we've pulled off of glucose to make that ATP. So we're transferring that energy from glucose into ATP. And that's our last step. But that's remember, our big bang for our buck, where we get all of the ATP. So to set the stage for cellular respiration, we have to look a little bit more about the parts of the mitochondria to kind of help us realize where this is taking place inside the mitochondria and why the structure of it is important. So there's an outer membrane and an inner membrane in the mitochondria. So the outer membrane is kind of like the shell, And then the inner membrane is going to do this kind of wavy pattern called the crista. And then the matrix is in between all those folds of the inner membrane. And this creates an intermembrane space. And so along this membrane is where a lot of this is taking place. So just keep this in mind a little bit as we go through the stages of respiration and where this might be happening within the mitochondria. So we have a couple of players in cellular respiration that are going to become very important. So, and these are our electron carriers. So we said that the cell is using those electrons that are falling off glucose. And so we have to have somewhere for those electrons to go. And that's going to be NAD and FAD, or how we term NAD and FAD. So NAD and FAD, kind of think of them like these little electron carrier dudes that pick up the electrons, and they are considered coenzymes. Okay, so remember we talked all about enzymes and coenzymes, and what they do is they accept those electrons. So they become reduced. So they grab those electrons, they become reduced, because when you gain electrons, you are going to be reduced. And so NAD becomes NADH, right? Because we've gained a hydrogen, we've gained an electron. And FAD becomes FADH2. So they each gain electrons. Okay, and what they do is they carry those electrons to the electron transport chain, and that'll make more sense when we go through that last step of the phosphorylation, the oxidative phosphorylation, and they drop them off. So essentially they're just carrying those electrons across the mitochondria, and then they drop them off, and they then become oxidized again. So then they turn back into NAD and FAD, and then they can go back, pick up some more electrons. So they're just the carriers. They just go back and forth, carrying those electrons, being reduced and oxidized. And that's going to become important, okay? So just keep NAD and FAD in the back of your mind as we talk about cellular respiration. So as a review, just go ahead and pause the video if you want to. A reduced molecule has lost or gained electrons, whereas an oxidized molecule has lost or gained electrons. So hopefully you're able to figure this out, but I know a lot of us haven't really covered redox reactions before, but something to keep in mind is LEO, or Leo, says GER, G-E-R. Okay, so that's a saying to keep in mind when you're talking about redox reactions, because when you lose electrons, you become oxidized. When you gain electrons, you become reduced. Okay? So a reduced molecule has gained electrons, whereas an oxidized molecule has lost electrons. Okay? So Leo says GER. Just keep that in mind, and that'll help you out, okay? So now let's start going through those steps of cellular respiration. And the first one is glycolysis. So this is really the first part in breaking down glucose. And what we produce is something called pyruvic acid, okay? And so by breaking down glucose into pyruvic acid, we actually get a couple of ATP molecules, but what we also get is some NADH. Remember those guys that are the electron carriers? So this is really important. So if we break down glucose, right? So essentially we're splitting glucose in half, essentially, right? Because we have 6 carbons, and then we turn it into 2 3-carbon molecules. You're going to get some ATP out of that because you're breaking bonds. And these electron carriers are going to pick up some of those electrons that fell off and become NADH. Okay, so we have filled up some of our electron carriers. So we get 2 ATP and 2 NADH per molecule of glucose in glycolysis. And remember, we have to keep in mind that glycolysis is happening outside the mitochondria, okay? In the cytoplasm, or actually cytosol, okay? Or just outside the bacteria if it's a bacteria doing glycolysis. So then our second step is going to be pyruvate oxidation. So now remember, we've just made 2 pyruvic acid molecules from glucose. So now what are we going to do with those pyruvic acid molecules? So this is our second step in breaking down glucose. And it does now take place in the mitochondria. So we have pyruvate, okay, which is another term for pyruvic acid. Okay, so we take pyruvate and we bring it into the mitochondria, okay, and we turn it into acetyl-CoA. Okay, and acetyl-CoA is a 2-carbon molecule. Okay, so what happens is so let's count our carbons, right? Because this is going to help us out. So if we have 2 3-carbon molecules, we still have 6 carbons coming in, right? Now, if we're going to break those pyruvates into 2 acetyl-CoA molecules with only 2 carbon molecules each, that's only 4 carbons. So what happened to our other carbons? That's going to be carbon dioxide. So we're already producing a waste now, okay? But we did make some more NADH. Okay, so we got another NAD in there to pick up some electrons that are falling off. Okay, and we've energized those guys to go to the electron transport chain. So we don't get any ATP out of this, but we do charge up those NADs, and we get some acetyl-CoA. And acetyl-CoA is going to be very important because it's what's going to go into the citric acid cycle. Okay. So now the third stage is the citric acid cycle, and this is the third and final step of breaking down our glucose. So we started with that 1 glucose molecule, we created 2 pyruvates, then we took those 2 pyruvates and created 2 acetyl-CoAs. We lost some carbon dioxide along the way, we've gained some ATP, we've gained some NADH, and now both of these 2 acetyl-CoA molecules are going to go through the citric acid cycle. So again, it's taking place in the mitochondria, and it's a continuous cycle. So it's always going, it's always turning over acetyl-CoA. And so we're going to completely break down acetyl-CoA. And we net 2 ATP. Okay, so 1 ATP per acetyl-CoA, but we have to remember, right, there's 2 molecules, two acetyl-CoA, so we're doing this twice. So since it's only a 2-carbon molecule, we're going to break it down and get 2 carbon dioxides, right? That's where our carbon goes. We're going to fill up some more NADH, so we have 3 of those. We get 1 ATP, and 1 FADH2, which is our other electron carrier. And so we have to do this twice, right? So for each, we're doing 2 cycles, so we have to times this by 2. So we total, with the 2 acetyl-CoAs, we get 2 ATP, 6 NADH, and 2 FADH2s, okay? Because remember, we have to double it. Okay, so let's review these stages so far, what we've got going on. So this is per glucose molecule. What are we getting? So first column is the stage, okay, where it's happening. I have a little mitochondria next to it if it's happening in the mitochondria. And then the second column is how much ATP do we get, how much NADH do we get, and how much FADH2. Okay, so for glycolysis, we get 2 ATP, 2 NADH. Pyruvate oxidation, we get no ATP, but we do get some more NADH. And then the citric acid cycle, we get quite a bit, right? We get 2 ATP and 6 NADH, and then we also get those 2 FADH2. So then our totals at the bottom of all of that so far, right? So this is what's happening so far with, we completely broke down glucose, right? Now you might say, okay, well that's not a lot of ATP, but we can't forget, we have all these stored up electrons, right? So now what do we do with those electrons? So now we have reached our final stage of glucose metabolism or cellular respiration. And this is what happens with all those electrons. And this is why those electron carriers are so important, okay? It's like putting money in the piggy bank. And this final stage requires oxygen. Okay, so this is very important because we'll talk about what happens when we don't have oxygen. And that's also why, as a living being, we all need to breathe in oxygen to supply our cells with oxygen to undergo oxidative phosphorylation. So there's 2 steps in the oxidative phosphorylation. The first is the electron transport chain. So that's where our electron carriers, NAD and FAD, are going to bring those electrons to the electron transport chain, and they get carried across the membrane. So this is where we talked about the membrane inside of the mitochondria. So there's an inner membrane that folds back and forth, so it increases the surface area for all this oxidative phosphorylation to happen. Okay, so if we look at our picture, we have an outer membrane out here, and then we have an inner membrane, which is where all the action is happening, and we have an intermembrane space and a mitochondrial matrix, which is kind of inside the folds of those inner membranes. Okay, so our second step is going to be chemiosmosis. Okay, I know that's a weird word, but we've kind of seen osmosis before. Okay, and chemi is chemical. Okay, chemiosmosis, and we're going to make, this is where we actually make the ATP, and we use an enzyme called ATP synthase. Okay. So this is how we're going to actually make that ATP. So we have 2 stages. We bring in those electrons for the electron transport chain, and then we're going to utilize that to create the ATP using this enzyme, ATP synthase. So here's our picture blown up. So we're going to walk our way through this, and I have all the steps laid out on the next slide as well. But let's just look at what's happening. So first, we're going to undergo the electron transport chain. So this is where our friends NADH and FADH2 are going to bring those electrons to the electron transport chain. So they're going to drop off those electrons, which are going to fuel these little pumps. Okay, these are the electron carriers. Okay, these guys are going to pump the hydrogen that's coming off the NADH, because remember, we're going to lose that hydrogen, lose that electron, and turn ourselves back into NAD+, just NAD, right? Same with FAD. So these guys are losing electrons because they're dropping them off, but they're also losing hydrogen, and that's the key. So we have all these hydrogen molecules over here, okay, because we're pumping all those electrons into these proteins. These protein complexes are then going to pump out the hydrogen into that intermembranous space, okay, against the gradient. Okay, so they're going to pump them all out. They're going to pump them all out that they don't want to go. And then what happens? Okay, So why we require oxygen is that the oxygen is going to bind the hydrogen left in this matrix, and then we're going to create water. But the big thing is we've pumped most of the oxygen out, right, into the intramembranous space, so it wants to come back in. So all these hydrogens out here are now going to rush back through the ATP synthase. And that is how we're going to create ATP, okay? We are going to make ATP using that ADP and the 1-phosphate, okay? And that's the chemiosmosis step. So that's our number 2 step, okay? So let's lay out those steps that we just went through, and then maybe it'll make a little more sense. Okay, so first, NADH and FADH2 drop off those electrons, but they're also dropping off hydrogens essentially as well, right? So those electrons go through the transport chain and pump all those hydrogens across the membrane. So the electrons are going to travel through, the oxygen is going to pull those electrons through to form water, using those hydrogens that are left over in the matrix, okay? And then because all those hydrogens are out in the intramembranous space, they want to dive back in to the matrix. So they're going to just diffuse through the ATP synthase, and that's what's going to drive the creation of ATP, okay? So this might be a little better picture in terms of what's happening with the electrons and the hydrogens. So the electrons are being dropped off and pumped through the electron transport chain, coming back over to oxygen, which will pick up some of these hydrogens, all the excess hydrogens that aren't being pumped through the membrane to create water. But all those hydrogens, all those extra hydrogens are all being pumped. These are hydrogen protein pumps, and they're putting them all out here where they don't really want to be. So then they're going to want to rush back through that ATP synthase, which is the enzyme that's going to create ATP. Okay? And this is where we get another 28 molecules of ATP per glucose. So we already had 4 going through our process of breaking down glucose, so that gives us 32 molecules of ATP. So that's quite a bit of energy. So cellular respiration allows cells to really create quite a bit of energy, but we have to have oxygen to undergo cellular respiration. So what happens if we don't have energy? Well, we undergo what's called fermentation or anaerobic respiration. So anaerobic means without oxygen, okay? So this allows some cells to be able to make energy without oxygen. You're not going to make quite as much, and there's going to be some penalties probably, okay? So what happens? So we only get glycolysis. So essentially, we're going to just do glycolysis over and over and over and over and over again to get ATP. So if we look at our little map, we take our glycolysis, right, which gives us 2 ATP. But if we have oxygen, we can get a whole lot more ATP because we undergo cellular respiration. If we don't have oxygen, we undergo fermentation, but we only get 2 ATP, but we can do it over and over and over and over again. But if we keep doing glycolysis over and over and over again, what also are we producing? We're doing 2 NADH per cycle. Now, if we keep doing this over and over and over and over again, and we're producing all these NADH, we're going to run out of NAD+. We're going to run out of electron carriers. So we have to get a way to recycle those electron carriers. And that's the process of fermentation. So there's two types of fermentation processes to allow us to recycle that NADH to NAD+, because we don't want to run out of those electron carriers or we can no longer undergo glycolysis. So there's two ways that these organisms have found out how to recycle NADH. So the first type of fermentation is alcohol fermentation. So a lot of us are familiar with alcohol, but maybe not how it's made. So some organisms, such as yeast, undergo alcohol fermentation to essentially come up with a way to recycle their NADH back to NAD+, so that they can continue the glycolysis cycle and continue making energy. So if we look, they undergo glycolysis, right? Glucose into pyruvate acid, which gives us our energy and our NADH, right? But we don't want to keep doing that or we're going to run out of NAD. So what they do, since they don't have any oxygen, is they turn that pyruvate acid into ethanol, okay? And I'll show you the process in a little bit, but you don't need to know the actual chemical reaction of how they do this, but just know that alcohol fermentation is one way to recycle our NADH to NAD+, okay? So the other type of fermentation is lactic acid fermentation. And this is done by our own cells sometimes, okay? When we have low oxygen, this is what our muscle cells do to continue going. Also, some bacteria do this as well, okay? So this is how they come up with the idea to recycle their NADH. So again, we go through our glycolysis and produce our two pyruvic acids. And what the bacteria in our muscle cells do is they turn that pyruvic acid into lactic acid. And I'm sure many of us have suffered from lactic acid buildup. You push your muscles too hard, low oxygen, and you get lactic acid buildup, you get muscle cramps, right? So this is when oxygen is low or unavailable, our cells undergo lactic acid fermentation, okay, to continue getting energy, but they have to recycle that NADH. So what do those processes look like? So essentially there's three fates for that pyruvate or pyruvic acid, which we get out of glycolysis. So of course our aerobic oxidation is the one that we use in cellular respiration. Well, we also have these two other options essentially, and they're both anaerobic, meaning no oxygen. So we have our lactic acid fermentation on the left, and essentially we turn that pyruvate into lactate, okay? And that allows us to recycle and become NADH back into NAD+. Okay. Now, alcoholic fermentation, we turn that pyruvate into ethanol or alcohol, right? So it's a little bit of a longer process, but we still recycle that NAD+. Okay. So this is how certain organisms and our muscle cells have come up with a way to continue getting energy without oxygen. But again, you do get some problems, right? So lactic acid buildup might not be good. Maybe ethanol might be a benefit. Well, we have turned it into a benefit for baking and beer and wine making, right? So I want you guys to know these three fates of the pyruvate. So let's do a little review at the end and pause your video and answer these two questions. What is the purpose of fermentation? And what are the two types of fermentation? So fermentation is also known as anaerobic respiration, right? So we don't have any oxygen, but we still want to produce energy. But we have this problem of NAD, right? So we've produced all this NADH, but we need to recycle NADH to get NAD plus again. Okay, so that's the whole purpose of fermentation, is to recycle our NADH. And what are those two types to be able to get NAD plus back? We have alcohol fermentation, which turns pyruvate into ethanol. And then we have lactic acid fermentation, which turns pyruvate into lactate. Okay? So what happens if we don't have sugar? So we talked all about glucose being broken down through cellular respiration, but what happens when we eat protein and fat? We can use that for energy too, right? Well, we have a preference, our body has a preference, and that is glucose first. But we can use fats and then we can use proteins, okay? So if we look at this picture here, and you don't need to know exactly the steps where fats and proteins come into play, but you can see that fats and proteins can be broken down into their components, right? Just like our polysaccharides are broken into our simple sugars, we can use fats and turn it into glycerol and fatty acids, which can enter the cycle at a couple different places. And then we can continue through the cycle to create energy. And then we can break down proteins into amino acids, and certain amino acids can enter the cycle at certain places. So again, we can utilize our other organic compounds as energy, but obviously our preference is going to be a carbohydrate, which will turn into sugar because you get the most energy out of it.